In this video, we will be discussing the structures of the prokaryotic and eukaryotic genomes. The typical prokaryotic cell will contain a single circular chromosome. Cells that have a single copy of a chromosome are known as haploid cells. The chromosome of prokaryotic cells is located within a region of the cytoplasm known as the nucleoid. The DNA of the chromosome must be packaged to fit within this region. Packaging of the DNA in bacterial cells is accomplished by folding the DNA into loops that are approximately 50,000 to 100,000 base pairs long, using proteins to hold the loops in place. An enzyme known as gyrase causes additional folding and supercoiling of the prokaryotic chromosome into a compact mass. This packaging is important when we consider bacteria such as E. coli that have a chromosome that is approximately 1,000 times longer than the entire cell. Following folding of the E. coli chromosome, the genetic material fits into a space that takes up about 10% of the cell's volume. Prokaryotes may have additional pieces of genetic material known as plasmids present within their cells. Plasmids are small circular pieces of DNA that are a fraction of the size of the bacterial chromosome that are capable of replicating independently of the bacterial chromosome. Plasmids code for structures or products that are not necessary for normal growth. In other words, if the plasmid were removed, the bacteria could still continue to grow. In many cases, the plasmids code for products that give the bacterium a selective advantage over other bacteria. Plasmids are described based on the types of products provided by the plasmid. The F plasmid, or fertility plasmid, is a plasmid that contains the gene necessary to produce pili. If you will please recall from Unit 1, pili are hollow structures used by some species of bacteria to transfer copies of their genetic material to other bacterial cells. The transfer of genetic material through pili is known as conjugation. F plasmids, therefore, are necessary for bacteria that perform bacterial conjugation. We will discuss bacterial conjugation in more detail in another video later in this unit. The R plasmid is the resistance plasmid. R plasmids, as their name suggests, contain genes that allow the bacterium to resist antimicrobial drugs or chemicals. We will discuss some of the mechanisms of resistance conferred by R plasmids in Unit 4. Bacteriocin plasmids contain genes that code for products known as bacteriocins. Bacteriocins are proteins that are toxic to other bacterial cells of the same or similar species that do not contain the plasmid. Those containing the plasmids have resistance to the toxin and are therefore protected. Bacteriocin plasmids allow bacterial cells to kill competitors. Virulence plasmids contain genes for structures and products such as enzymes and toxins that allow a bacterium to become pathogenic. We will be exploring a number of virulence factors in Unit 3. Eukaryotic genomes contain both nuclear as well as extranuclear DNA. We will examine both. Nuclear DNA refers to the DNA located within the nucleus. Eukaryotes typically have more than one chromosome located within their nucleus, and the chromosomes are linear. Many eukaryotic cells are diploid, meaning that they have two copies of each chromosome, one paternal and one maternal. Eukaryotic chromosomes must be packaged to fit within the nucleus. Eukaryotic DNA is packaged using proteins known as histones. DNA has a negative charge which is attracted to the positive charge of the histone proteins. DNA wraps around the histone proteins to form structures known as nucleosomes. When the cell is not dividing, the DNA and its associated proteins exist as chromatin, a thread-like mass of DNA. The nucleosomes clump together with other proteins to form the chromatin fibers. The chromatin is loosely packaged in areas of the chromosome that contain active genes. The areas containing active genes are known as euchromatin. Areas of the chromatin that contain inactive genes are more tightly packed and are called heterochromatin. During cell division, the DNA and proteins are condensed to form rod-like structures known as chromosomes. The extranuclear DNA of eukaryotic chromosomes includes the DNA found in chloroplasts and mitochondria, as well as the plasmids found in some fungi, algae, and protozoans. The DNA found in chloroplasts and mitochondria resembles that found in bacterial cells. It is a single circular chromosome. The genes located on these chromosomes encode for some of the proteins needed in the processes that occur in each of the organelles. Mitochondrial DNA is responsible for coding for approximately 5% of the RNA and protein molecules needed for mitochondrial function. The cell's nuclear chromosomes code for the remaining 95% of the RNA and proteins needed for mitochondrial function. The plasmids found in eukaryotic cells do not appear to play a role in adaptation as we see in bacterial cells.